My talk will be quite molecular in nature. We're going to go a little bit into depth of virus cell interactions from the molecular standpoint. So there's not going to be much clinical or anything like it. Anyway, so with that, I'll first of all would like to talk a little bit about what I'm going to talk in general about. So what I'm going to do is I'm going to first make a few comments about the importance of lipids in HIV replication cycle. Then I'll need to go a little bit in depth of the principles of superposition fluorescence microscopy techniques that are used in the lipid behavior studies of HIV-1. This is important for you to understand exactly what the data that I'll be presenting afterwards represents. And then I'm going to present some results from my study on lipid behavior within monocyte virus containing compartments. But before, of course, I'll introduce the background to the topic as well. So, first of all, a little bit of an introduction, and I'd like to start with, I guess, a bit of a bolder statement that I believe that actually comparing to the proteins, the role of lipids in the biological processes is generally not well studied. Why is that? That is generally because, well, lipids are more difficult to manipulate and observe in the first place. We have, I think, better reagents in terms of how, when observing proteins than lipids. Generally, also, when you stain lipids, lipids appear quite homogeneous in nature. Generally, when you observe proteins on a cell, they form distinct structures. Whereas with lipids, you know, you stain the membranes and everything appears quite homogeneous and not so interesting. However, thanks to recent advances in technologies and generally, there have been actually quite interesting studies performed by me and others, where we studied lipid dynamics in HIV replication cycle. And actually, those studies have revealed quite unique mechanisms that have shown how lipids influence virus assembly and entry. I am not going to go into too much depth because that isn't the topic of this presentation. But for example, we're able to find out that there, on the surface of virus particles, there's quite an unusual lipid behavior. And that in turn, for example, influences how HIV-N clusters. And obviously, then in turn, that HIV-N clustering is very important for the virus entry. On the other side of the virus replication cycle, we found that there is actually quite previously unappreciated or unknown fact that rather than utilizing lipid rafts for virus or cholesterol-enriched domains for virus assembly, HIV creates those domains by itself. It's actually HIV GAG is remodeling the local lipid environment at the virus assembly sites, rather than utilizing existing ones. So these studies were actually possible thanks to the advances in fluorescence microscopy techniques, which those studies utilize. And the very important point of these techniques was that they enable us to observe lipid dynamics, also protein dynamics as well, if you want to, but at the scales of individual virus particles, whereas this was not possible to do before. So with that, I'm going to introduce several concepts which relate to how those techniques works. First of all, I'd like to draw your attention to the fluorescence microscopy resolution limit. For all those of you that work with microscopes may know that when you actually try to label something, in this case a single virus particle, for example, here I show that the virus particle here is, for example, labeled for ENV. What happens when you try to image it via a standard confocal microscope, for example? You have your excitation beam, then you read out the signal. However, what you get is diffraction limited image, which is much bigger than the virus itself. And the resolution limit of the microscope is generally 250 nanometers in the XY plane and Z is 600 nanometers. So whenever you try to visualize a virus by itself, you get a smeared out dot. So the blue circle represents the true dimension of the virus, whereas this represents several molecules of, in this case, ENV molecules labeled on the surface of HIV, and you get a big blob, which is bigger than the virus itself. And this makes it impossible for us to access any information below the size of 250 nanometers, or in this case, the size of the virus, which is about 100 nanometers or so. However, there have been quite a strong advances over the last 10 years in terms of super resolution microscopy, and that microscopy with super resolution means that we can go beyond the resolution limit. One of those techniques is stimulated emission depletion microscopy, or STED. And this microscopy technique simply, without getting into too much detail, modifies the confocal microscopy with a second additional laser called depletion laser. And otherwise, otherwise it's very similar to confocal microscopy. But when we excite the fluorophores by a normal excitation beam, the green one, we also apply the red depletion laser. And in a nutshell, what does that do is it squeezes the excitation volume to much smaller diameter. And this allows us to observe things at much higher resolution. And so here for super resolution microscopy of this type, we reach about 20 to 40 nanometers in XY direction. So when you compare the images of the same virus taken in a confocal mode, this is an N of molecules in confocal mode as I showed earlier, and we look at those in a super resolution mode, now you can see that we can actually detect individual clusters of N on the surface of the virus. So this gives us access to the information information from microscopy through microscopy that we did not have before on the scales that we couldn't before work with. So that is one concept. Another concept that I need to introduce is the concept of fluorescence correlation spectroscopy. And as I said, the studies of lipids often require you to look at lipid dynamics. And how do we study those? We utilize a fluorescence correlation spectroscopy, which monitors the fluorescence fluctuations of lipids as they pass through the excitation volume, the laser excitation volume at the microscope. And then you just simply have a readout of the fluorescent, as fluorescent molecules pass through the excitation volume, you read out the fluorescence intensity. Then you perform autocorrelation curves on this without getting into too much mathematical details. You convert these fluorescence fluctuations into the correlation curves. And what this correlation curve gives us is some very useful information about the behavior of the molecules that are interrogated, be it proteins or in this case lipids. So we can find out information about number of molecules that are transiting through that excitation volume, single excitation volume, and also very importantly, we have information about average transit time across this excitation volume of the microscope. And with average transit time known, we can get a molecular diffusion coefficient and also get molecular mobilities, and thus get molecular mobilities of the molecules in question. So how does this tie into the STED microscopy? Well, normally FCS is also diffraction limited. As I said, the FCS by itself is also a standard technique, so it's bound normally to the limits of standard microscopy. But you can now combine STED with FCS. So whereas in confocal, the excitation volume is approximately in size of 250 nanometers, and that is bigger than a single virus particle. With STED combined with FCS, we can now observe things, the dynamics at the scales below the size of individual virus particle. Taking this step, taking that concept even further, we can combine STED FCS in so-called scanning modality. And that basically allows us to retain some of the spatial imaging with spectroscopy. Just going back one, fluorescence correlation spectroscopy only focuses always on a single spot. There is information only about a single spot that you're focusing on. Whereas with scanning STED FCS, we gain some spatial component to it, which means we can interrogate several different physical spots all at once at the same time. So the way it works is we're scanning the laser beam across several different positions. Either we can do it via line or an orbit. And each one of those positions, then we get information about fluorescence fluctuations. We then get correlation curves and get molecular mobility about every one of these spots. And why is that useful? I'll show this using an example of the previous study. For example, we utilize this precisely to look at the lipid mobility of fluorescent lipids inside and outside of a virus assembly site. So here is a cartoon showing individual virus assembly site. And we interrogated in orbit scanning, we have interrogated areas outside as well as just inside virus assembly site. Here is a fluorescent image showing a single virus assembly site. And And we, when scanned dynamics of lipids inside and outside, we're able to find out that certain lipids get trapped as the virus begins to assemble, whereas other lipids do not. And this was quite novel. This is quite a novel approach and giving us information that was, not known before. But I'm just using this as an example, or a practical example of a study. And now I'd like to go to the background of the study that I want to talk about, that I'm talking about here today. So I need to introduce now to tie it in with what is Siglec-1, what are virus containing compartments, what is transinfection. So basically, from the top, Siglec-1 transmembrane protein was found to be an essential molecule that is involved in capture of HIV by mature dendritic cells in a process called transinfection. So on the surface of what is Siglec-1? Siglec-1 is able to trap virus particles via the gangliosides that they exhibit on the surface. And this process is generally non-specific. The Siglec-1 is able to capture ganglioside of anything that has that displays them, be it HIV or any other viruses. But what it does with them is then it sequesters those upon capture, sequesters those virus particles in the specialized compartment called virus containing compartment. And not much is known about this virus containing compartment. But why is it important? This compartment is important for the process of transinfection. So the idea being is that the mature dendritic cells, which by themselves are not infected, are not infectable by HIV, they capture HIV virus particles from infected T cells. They capture them and they sequester them, and then those virus particles in this virus-containing compartment are then available for transfer into another T cells that are interacting with mature dendritic cells. In addition, those virus-containing compartments in mature dendritic cells act as a long-term reservoirs of HIV in the infected people. So, obviously, the rationale behind this is to try to find ways to combat these long-term reservoirs. And to do that, we need to find out more about the virus-containing compartments. Of course, there have been some nice advances recently in our group. It has been shown that there are some quite good antibodies. Anti-Siglec-1 antibodies are able to block this interaction, and they are quite potent antiviral compounds acting against HIV, as well as, in this case, also Ebola virus particles. But the problem with this approach is that the VCCs themselves are not accessible to antibodies due to the very labyrinth-like nature of the VCCs. They're not accessible, and thus the antibodies present only a part of the solution to combating HIV reservoirs. So this is the rationale, as was mentioned a moment ago, a rationale behind this VCC lipid behavior study. Although we can block the transinfection via anti-Siglec-1 antibodies, this doesn't block does not act on the level of VCCs. And so we need alternative strategies to combat these HIV reservoirs. And firstly, we need to find out something about those VCCs. Not much is known about them. So what I thought when I initiated this study is to take this knowledge that I have of being able to look at the dynamic behavior of lipids, and knowing that these virus-containing compartments have a very labyrinth-like structure, hypothesized that they may have a different dynamic behavior of lipids inside VCCs. And this is the first objective of my study. Basically, I wanted to establish the dynamic behavior of lipids inside VCCs and contrast it with the behavior on a plasma membrane to see if there is anything different about those VCCs. And then roughly, I wanted to see if there is any difference. If there is any difference, I wanted to see if I can interfere with those VCCs by utilizing drugs that block parts of the lipid pathways. and see if they do have if there is any effect on forming of the diseases. And the way I'm doing these studies is via microscopy imaging and via aforementioned scanning FCS microscopy technique. So first, one of the first steps that I did is basically to look at how do those VCCs look like under superposition microscope. So to talk about the method a little bit, what I simply am doing, the general pipeline is that I utilize interferon-stimulated primary mature dendritic cells, and I incubate them with HIV-1 VLPs, which contain GAGI GFP, which are made out of GAGI GFP. They're not full viruses, but as I said, the Siglec-1 capture occurs via gangliosides, and so you do not need a virus, entire virus, full virus for this to work. HIV VLPs will be just captured just as well as a full virus by dendritic cells and sequestered in those VCCs. Then what I'm generally doing is I pulse the live cells with fluorescent lipid analogs. In this case, I use phosphatidylcholine. This picture is phosphatidylethanolamin, but I also use cholesterol and sphingomyelin as well for my studies. I do a short pulse, and in this case, I fix the cells and I stain them also for Siglec-1 using anti-Siglec-1 antibody. So on the left side, we have just an overview of showcasing several cells. In the green is virus particles, purple magenta is Siglec-1 signal, and phosphatidylethanolamin is in orange. And here, just on the confocal overview, we see those virus containing compartments, and they represent, they basically are those really, really bright areas. In those bright areas, there is a lot of virus particles. There is Siglec-1, and what when we zoom in here, you can actually see in purple Siglec-1, and also in green individual virus particles. And on the right side, this is a Z-stack. This is a three-dimensional Z-stack showing a cut through across those virus-containing compartments. And we can see that they're actually quite complex structures, full of virus particles. So there is quite a complex, they are quite complex in nature and full of virus particles. What was interesting to me that I found out is that they seem to also incorporate quite a lot more of lipid molecules, and this is irrespective of whether I used cholesterol, or I used phosphatidylethanolamin, or any other on a lipid probe. It seemed like even when I'm pulsing the cells with very little lipids, and that is necessary for good fluorescence correlation spectroscopy measurements, it seems like those VCCs contain always sequester quite a large number of lipid analogs. I'm not 100% sure, but I believe that this is simply due to the fact that those compartments are quite compact and complex. They have complex morphology, incorporating a lot more lipid molecules in a given area. So that would be my explanation, but I am not 100% sure why is that occurring. However, this already indicates to me that this compartment is quite interesting and behaves a little bit differently from a plasma membrane. So then, when looking at even those static images, it occurred to me that the studying of VCCs via fluorescence correlation spectroscope will be a quite difficult endeavor. Well, why is this? Because of this very convoluted and dense virus-containing compartment. This really necessitate me using very as high resolution as I can possibly do. The problem with if you're utilizing confocal approach, for STED FCS, this is, for example, here on the top, those spheres are excitation volumes. If I utilize just a standard FCS approach, then I'm running into a danger that I'm gonna look at the several different membranes all at once, including a plasma membrane as well. So I need to utilize, and this will confound my results, because if I'm going to start acquiring multimobility of lipids from several different membranes, including the plasma membrane, this will influence my results. So what I really need to do is I need to squeeze this excitation volume as much as I can, and I also need to need to make sure that I'm operating very much away from plasma membrane, and to capture as little slice of that membrane as possible, especially given the fact that I believe that those membranes are quite convoluted. So multiple membranes stacking on top of one another may interfere with my results. So this is one of those caveats that made me realize that this study will be quite difficult. I'll need to do it, by hand, manually selecting those VCCs for observation very carefully. Another issues were, of course, you have the usual cell movement. We are operating in live cell imaging, So there are all these sort of problems. In addition, there may be a potential effect for VLP's membranes. However, from my experience, I found out that the VLPs do not, or virus particles do not really like to incorporate exogenous lipids due to their very rigid lipid structures. So that is not much of a problem. And like I was saying, the membranes themselves are quite problematic, but I stuck on, and for quite a long time, I basically very slowly acquired the data on the behavior of lipids. So how did I go about this? Once again, I started with interferon stimulated cells. And here I have to actually make a disclaimer that because of such a slow going of acquiring the data, in the end, I had to move on to something simpler. I could not use mature dendritic cells, primary cells, because the process of procuring them is quite slow. Is to prepare them takes a long time, and I needed a simpler system. So I moved to the THP1 monocyte cells, which replicate the aspects of the dendritic cells virus-containing compartments. So they also create those, but they're much easier to work with, obviously, because it's a cell line rather than primary cells that need to be extracted and matured and stimulated. Otherwise, my protocol is pretty much the same. I incubate the interferon-stimulated THP1 cells with VLPs. I label them with a fluorescent lipid, phosphatidylethanolamine, or cholesterol, or sphingomyelin. And then I look at try to find the VCCs. So here is an example of overview of a single cell on the left. Orange is the lipid, and the green represents virus particles. Then what I do after seeing something that looks interesting, I zoom in a little bit more, and you can see in the middle bit of a zoom of the virus-containing compartment. That little sac here represents, I think, a virus-containing compartment with the virus particles lining its walls. I can already see that I'm well away from roughly in the middle equatorially from the cell, so I'm well away from the plasma membrane. And then what I do is I focus on a very small portion of that virus-containing compartment. I often try to target the area just next to an individual virus particle. And then what I do is, like I was showing before, I acquired so-called the correlation carpet, fluorescence intensity carpets, which then I convert into the autocorrelation functions. And this gives me the autocorrelation function dependent on the position of that line where I'm scanning for mobility. And I repeat this process many times, effectively trying to find out the best curves for both plasma for VCCs and the plasma membrane. And this quite a lot of work can be well summarized in just several graphs. And it's kind of sometimes funny that a whole year worth of work can often be summarized into just 2 graphs. I have data for cholesterol and phosphatidylethanolamine. I was working with sphingomyelin. However, unfortunately, I found out that the molecular probe that I was using for a long time, in the end, does not replicate the proper behavior of sphingomyelin. Speaking with my colleagues in biophysical field, that it's quite difficult to find actually a sphingomyelin probe that behaves correctly. And this actually is responsible for quite a lot of different results that people in different studies had whenever utilizing this probe. So in the end, I had to drop it, and I may in the future try to procure a probe, which is difficult to get, but maybe a little bit better. This is one of the caveats why lipids are much more difficult to study than proteins, because with proteins you have antibodies, but with those probes, fluorescent lipid analogs often simply do not behave as lipids should. Anyway, to the results. And so after quite a long study, I have actually found out that if I compare the cholesterol behavior between the plasma membrane and behavior of this lipid within VCCs, what I found out that cholesterol is much more trapped has much more trapped behavior than one on the plasma membrane. And this does not seem to happen with the phosphatidylethanolamine. As phosphatidylethanolamine is a more of neutral lipid, whereas cholesterol, as you know, is involved in liquid order domains or lipid rafts, and its trapping is often indicative that the membrane has quite different properties. It's more rigid, it's more confined, and it's trapping completely different lipids than what we could consider a normal plasma membrane. And this is very interesting because, as we know, VCCs are contiguous with the plasma membrane. They are like very complex invaginations. Nonetheless, they are of the same surface. Yet inside this compartment, the lipid behavior is quite different, which is quite interesting. So then, with this well, quite actually encouraging result, I moved to the testing of whether this has any practical applications, and then I utilized this knowledge that the VCs have a very different molecular mobility, so lipids behave differently with VCCs, and I tried to interfere with this process. And what I've done here is I have utilized interferon-stimulated THP1 cells, and I have used two drugs for start off. These two drugs, LB1 and DNJ, are interfering with de novo sphingolipid synthesis, and they block it. So what I did is I incubated the cells with those drugs for quite a long time to ensure that its dividing cells have progressively less and less of those sphingolipids, because those drugs block de novo synthesis. They do not deplete the existing sphingolipids. So you have to wait a while before the cells drop down in the amount of sphingolipids. And well, sphingolipids are also indicative of cholesterol-enriched domains or lipid rafts. So often enough when cholesterol is slowed down, also the sphingolipids are thought to play a role. The sphingolipids also play a role. And these were the drugs that I had on hand. And so I pushed on with this quite preliminary study. So what I did after those 3 days is first very important. I confirmed that the cells are equally viable via flow cytometry, so to exclude any issues that those drugs may induce apoptosis or change the cells, are toxic to the cells. And then I incubated them with HIV-1 VLPs as before. And here, via quite a simple approach, what I've simply done is I measured the percentage of the positive cells. So I grabbed a lot of random screens of cells on the cover slips, and I simply compared the number of cells that seem to contain those well-developed virus-containing compartments, which are characterized by very bright spots, blobs of green in the middle of the cells. And so when I compared a couple of hundred cells for each one of these and looked at the number of positive cells that are positive for VCCs, I found out quite interestingly, in this my first experiment was that it seems that one of the drugs seemed to reduce the number of VCCs quite a lot, but not another drug. So this is quite a preliminary result. I'm not sure how to interpret it, but it seems that this gives me another quite an interesting result. It seems that when we start interfering with the lipid balance of the cells without killing the cells, it seems that we're able to reduce. I mean, this indicates that we can reduce the number of VCCs, which actually is, I think, encouraging result, suggesting a possible avenue for treatments. I don't know yet, but the study, I think, needs to continue. But this is, I think, quite an interesting result in its own right. So with this, I will wrap up. And so first of all, what this study we have found out that HIV VCCs, unlike plasma membrane, display liquid odor-like liquid behavior that is distinct from plasma membrane. And I think the sphingolipid reduction appears to affect the ability of cells to form VCCs. Of course, there is quite a lot more things that can be done. I need to, first of all, we need to ideally confirm these results in mature dendritic cells now that I have the system worked out. Of course, with the last year, ongoing epidemics, my access to the microscopy was also reduced, and the number of experiments that I could do was also reduced, as we all know. And that was a problem. But now you think that I now have a better access to the microscope. I think now we would be able to work and confirm this in mature dendritic cells. And also the idea would be to try, try more drugs which interfere with lipid synthesis and or behavior and interfere with those. One of these drugs may be peridifosine, which is actually a drug which is tested in the cancer therapy. But this drug is known to actually disrupt cholesterol-enriched domains. I'll be very curious to see if that may have any effect on the virus-containing compartments. So there is quite a lot of work that can still be done. These are interesting results, but I think it opens up quite a lot of interesting avenues to go on, and also it provides yet another little piece of the puzzles, learning a little bit more about virus-containing compartments. So with that, I will stop here, and of course, I'd like to acknowledge my host group, Javier Martinez Picado group, and from that group, I'd like to thank Itzia and Xavi, who helped me last time, giving me access to mature dendritic cells and helping out in the laboratory. And of course, I'd like to thank Nuria as well, who now has her own lab in IrsiCaixa, because she actually introduced me to this topic, and through her, I was able to start this study in the first place and apply for funding. And this project has been funded by Marie Curie Skłodowska Actions Fellowship. And with that, I will stop and I will take any questions that you may have. Thank you very much. I'm not sure how this works, but if somebody wants to say something, either they can raise their hands, or they simply activate the microphone.
Yeah, just one thing, Jakub.
Yes.
Creo que tenéis acceso a activar los micrófonos si necesitáis hacer alguna pregunta, o podéis utilizar el chat. Maybe with the chat or the microphone, but we can wait 1-2 minutes.
Yeah, absolutely. Let's see if there is any questions. If you have, of course, any questions afterwards, if you think of some questions afterwards, my email was at the bottom, so feel free to contact me, of course, if you don't have any questions right now.
So Jakub, can you hear me? This is Javier.
Yes, I can hear you. Hi, Javier.
Nice presentation, Jakub.
Thank you.
Couple of questions I had through your presentation. One of them is, we know that these BCCs are, as you said, membrane invaginations of the plasma membrane. And from some data that's been produced by electron microscopy, apparently these invaginations can be really deep into the cell cytoplasm. So my question would be, how far by STED microscopy can you dive into these BCCs if they are far away from the plasma membrane? It's my understanding that you might have some limits in how deep you can go into the cell. So my first question is about that, how close, how far. You said that you don't want to be too close to the plasma membrane, but you cannot be too far away from the plasma membrane either. So
Precisely.
what's a compromise?
Exactly. It is a very, very careful compromise that I have to take, because the deeper I move away from the cell cover slip and from the cover slip of the microscope slide and the plasma membrane, the worse my resolution gets. So this is actually another caveat that I had to contend with, that I had to find out those VCCs which are as close to the plasma membrane as possible and as well nicely looking, but also at the same time not overlapping very much with the plasma membrane. I can of course go within a micrometer or 2 micrometers deep, but I wanted to find out those VCCs which are very, very close. For imaging, it's fine. You can, the imaging is you can go quite deep. I would be able to go into even on a tissue, look at this in a tissue level. But when I'm doing these dynamic studies, when everything is live, everything is moving, those are very, very sensitive to any optical aberrations. So I am very, very much confined to, as I said, like you said, Javier, this compromise. Be just a little bit away from a plasma membrane, but not too deep into the cell, because even a few micrometers deeper into the cell, already I'm gonna get worse results.
And my second question is regarding to the sphingolipid inhibitors that you've been using and we had used before as well in other experiments. I mean, the data is kind of surprising. I agree with you, because FB1 and DNJ are part of the same metabolic pathway inhibitors, but let's say that DNJ should be more specific of glycosphingolipids, while FB1 would be more like the whole ceramide metabolic pathway. So I agree with you. I don't know how to, explain these observations. My only concern, and maybe you have some insights into that, is whether the concentration of compound may be playing a role, because as I think that you just use one concentration, maybe it's an issue of concentrations. I'm not sure, because we tested these compounds for other purposes, not precisely to what you are doing now. So maybe, inhibitor concentration may play a role there as well.
I totally agree. This was it was one of just first experiments that I did on this. I utilized, I think, what was considered as in a quite high dose, but simply this may be a question of it not working. So definitely I'll need to repeat this few more times, because you're right, I was quite surprised why one compound does have an effect or not. Although DNJ, I know, has, I think, a couple of other effects. It can be used for immunosugar inhibition and so on. So it's a little bit more complex than that. So also, it may be exactly the concentration, but it may be that the DNJ is working via some other mechanism, nothing to do, and it has actually nothing to do with the sphingolipid inhibition as well. So I have to test that, and ideally perhaps some other drugs as well. So yeah.
Did you have the chance to look in whether cyclic 1 is equally present when you use one or the other inhibitor?
Yes, because we looked at the equal. Actually, that's right. Sorry, I mean, maybe I did not make that really clear. What we've done is we look at the equal cell viability and also equal amount of cyclic 1 expression in the cell. Sorry, that's that's an important aspect. I didn't put that. I forgot to mention it. Yes, we looked at it, that the cells are equally viable, and that the cells are expressing the same amount of cyclic 1 using, and we did that using flow cytometry. Yes.
Thank you.
Thank you.
Okay, thank you, Jacob. If there is any more question, we will see you next Friday.
Great. Thank you very much. Thank you for listening.
Thank you.